Hi. I'm Michal from the Chi Lab at Caltech, and I work with Shahab a lot on that neuroblastoma project. I'll share with you a little teaser about a different project together with Louisa from the McMahon Lab at USC, where we used SECFISH to study kidney regeneration. And just briefly, my take-home message is here in this slide. Basically, we're interested in this reciprocality between gene expression and cellular environment. And what I want you to take from this talk is that fibroblasts showed the strongest neighborhood-dependent gene expression patterns. So just to walk you through the experiment real quick. So we use SECFISH imaging to quantify expression of 1,300 genes in kidneys. We were able to, is it moving? No, it's not. Do I need to press enter? No problem. Okay. We were able to call cell types, look at microenvironments. We were able to see that microenvironments change dramatically after the kidney is injured. And we were able to see that fibroblasts change their expression pattern depending on where they are, and find specific spatially localized signalings between fibroblasts and injured cells. And please come talk to me afterwards. Thank you.
Hello everyone, I'm Hyobin Kim. I'm a project scientist at Cedars. Recently, I developed the cell neighbor index, which is a Python package to investigate gene expression change caused by direct cell contact. Firstly, cell neighbor index can identify neighboring cell-dependent genes using spatial transcriptomics data. Secondly, it can suggest new potential genes involved in cell interactions, which are not captured by ligand receptor pairs. Thirdly, it can explain cellular heterogeneity. So in a nutshell, cell neighbor index provides a new perspective on cellular communication between two adjacent cell types. Our research was published in Molecular Systems Biology. It was selected as a journal cover paper. So if you have any questions, please feel free to contact me. Thank you.
Hi everyone, my name is Kruttika Dabke and I'm a postdoc in the Parker and Jones lab here at Cedars. And today I'm going to very briefly talk about the mutational landscape of ovarian cancer and how it defines the proteome, spatial organization of tumor stroma and immune cells. So we have profiled high-grade serous ovarian cancer tumors who have both homologous recombination deficiency or proficiency, meaning a lack of ability to repair DNA damage, and also tumors before and after they've been exposed to chemotherapy. And so we've done molecular profiling with proteomics, methylation, and transcriptomics data, as well as whole genome sequencing. And that has really led us, or that data results from that have indicated to us that the homologous recombination deficiency is driving the immune landscape in these tumors. And that prompted us to do some spatial profiling, where we use a combination of machine learning tool called HooverNet, which takes in H&E stained images and gives really these broad categories of cell types within the tissue. And then we added on the 26-plex immunostaining with Akoya's phenocycler fusion, where we obtained about 36 million cells from 23 different tissues. And overall, we had 7.6 million immune cells, which is shown here in as a representative figure. And majority of them are the highest percentages was M2 macrophages followed by M1 macrophages, which was expected. And then we saw differences in the cell percentages for between HRD and HRP tumors, where M1 macrophages were significantly higher in HRD tumors, and dendritic cells were significantly higher in HRP tumors. And then we were able to do the spatial proximity analysis, where each of these lines indicate the proximity of those immune cell types. And in general, we saw more immune interconnectedness in HRD tumors compared to HRP. And that has led us to this hypothesis that maybe there is more a coordinated immune response in these tumors. And we also have PD-1, PD-L1, Ki67 staining for these, and that's kind of our next steps. This is just 6 months into the data analysis. So we're really excited about this data and hope to get some more insights into it. If you have more questions, please feel free to contact us.
Hi everyone, my name is Nawel. I'm a graduate student in Student Coders Lab here at Cedars. So our lab is studying cripping fat, which is an extranasal manifestation of disease that is defined by the expansion of the mesenteric adipose tissue around the inflamed small bowel. And so we observed that in the cripping fat tissues, there are fat-associated lymphoid clusters, or FOCs. However, those FOCs are not present in the paired mesenteric adipose tissue. So we hypothesized that bacteria induce the FOC formation and colocalize with FOX immune cells in cripping fat. And so to study this, we did IMC on 12 paired cripping fat and MET samples from Crohn's disease patients. And we also did FISH using a probe targeting all bacteria. And so the IMC images showed us that the FOX in the cripping fat are heterogeneous, with some FOX showing B cells at the periphery with CD4 T cells in the middle, while other FOX have a mix of B and T cells with no specific spatial localization. And so suggesting that there are different developmental stages of the FOX. And then the fish showed us that the bacteria here are present inside the FOX. And so we also did a pseudotime analysis, which here showed that the MET samples cluster on the left of the trajectory. We have a cripping fat cluster at the very end of this trajectory here. And over here, interestingly, we could have a potential transition of the samples going from MET to cripping fat. And so then looking at what markers were expressed in this transition, we saw an hepatic marker as well as IgA. And then at the very end of the trajectory in the cripping fat cluster, we saw expression of CD4, for instance. And so overall, suggesting that during this MET to cripping fat transition, we could have lymphatics transporting bacteria, leading to IgA release, and then over time, an adaptive immune cell response with FOC formation in the cripping fat. And so same thing, if you have any questions, please feel free to let me after. Thank you.
We did have one more speaker who, I don't know if Colt is here. He had a meeting. Is he online? Probably not. Okay. So sorry, Colt, but if you do come back, I just yell into the camera, I guess.
Hello, I'm Reyhaneh, post-biochemical lab here at Cedars. Our project is about iPS-derived macrophage cells for cancer treatment. Traditional cancer treatment cause severe side effects, so we are turning to immunotherapy to change it. Actually, cancer cells can trick macrophage cells with don't-eat-me signal, so we are trying to block this access to make macrophage cells more effective. We use anti-CD47 to block this signal, and as you can see in this video, our iPS-derived macrophage cells can eat anti-CD47 treated lymphoma cells in less than an hour. Next, we evaluate their trafficking potential in vivo by using NSG mouse model for lymphoma, and we use a Lunaphore technology to detect our cells. We have been able to detect our IMAX trafficking into the tumor tissue 1 day and 4 days after injection. And we, at single cell resolution, we also could detect a shift in their phenotype and also metabolic status. And next, we will focus on modification techniques to improve their potential for cancer treatment. And that's it. Thank you. If you have any questions.
Hi, I'm Fakhar Singhera, and part of US. I'm a PhD student at USC and part of the Lee Lab at Health Institute. I'm going to talk to you about a model for sarcophagus using both healthy purchasable cell lines and patient-derived bone cells. Sarcophagus is essentially a disease held by diabetic people that causes a open wound continuously at the bottom of their foot, like an ulcer. It's thought there's no actual mechanism of initiation that is actually known. It's thought to be caused by an interplay between rank L and rank, and that causes a lot of osteoclast recruitment that continuously causes the bone to try to break down, and a little bit builds up again and it breaks down again. There's no actual model for it, so we were trying to figure out what the mechanism of initiation is. To try to do that, we've been looking at cytokines that are expressed and that cause tissue injury or recruit for tissue injury, recruit repair cells, and recruit inflammatory cells. We've been lucky enough to work with a surgeon. We can get actual bone samples from people's surgical from surgical patients throughout the foot, and also blood. And we've been putting that through like a platform that allows us to detect multiple cytokines and like how those are being excreted by the cells. one thing that we've noticed that's super cool so far is that patient cells, yes, they are going to display a lot more cytokine polyfunctionality, meaning multiple types of cytokines, much more than cell lines. But the further you get away from the body, the less it happens. And also, if within this cytokine platform you put a bunch of the cells together, like so you have multiple cells, they will actually increase. They'll have paracrine communication between each of the cells, and there's a lot more polyfunctionality, and so they're actually expressing a lot of different types of cytokines. If you have any questions, please feel free to reach out, and thank you.
Hi, everyone. My name is Sara Ancel. I'm a postdoc at Sanford Burnham Previs down in San Diego. And today I'd like to share with you our latest effort on the field of spatial glycomics. So I'm sure that you're very well aware that recently tremendous advances have been done in the field of spatial omics technologies. And most of that effort has actually been done by mapping the transcriptome and the proteome of tissues. However, approaches to kind of like gain spatial information on post-translational modification, including, for example, glycosylation, which is the addition of sugar molecules to proteins, has kind of remained scarce. And so to kind of tackle and to help tackle that need, so what we've done is that we have actually developed an approach, like a multi-parametric imaging approach to kind of enable the visualization of glycans within and given their specific locations within the tissue. And so basically that new sugar map can be overlaid on top of cell type markers. And so that is really for us to see the unique glycan features of different cell populations within a given field of view, but it also allows us to highlight some specific structures that cannot necessarily be visualized using only anti-base approaches. And because glycosylation is a non-template driven modification, it has one of the advantages that we can actually apply that method to different tissues and also, more importantly, multiple species. And so basically, because glycans have critical roles in development, disease, and communications between the cells, we believe that pinpointing exactly where glycans are located within the tissue can really help us gain another layer of understanding, basically, of tissue biology, and also, of course, insights into the impacts that glycans can have in health and disease. And with that, I'd like to thank you for your attention, and we'll meet at the break room. Thank you.
Thanks, everyone. Yeah, we're gonna have one more coffee break, and we'll see you back here in 15 minutes.